Good morning, all. Welcome back to our official YouTube channel, SRNBGNR Government Arts and Science College, Khammam, Department of Biochemistry. Myself, Dr. B. Satish Kumar, Assistant Professor in Biochemistry. In last few video lessons, you have been learning about some basic process involved in molecular biology. So this molecular biology paper is in the syllabus of final year students, 6th semester, 7th paper, core paper. And in this unit, so far we have covered the first unit. That uh, the syllabus of first unit contained replication and transcription, and as well as the post transcriptional modifications. And as far as the second unit is concerned, this unit, the syllabus in this unit is translation. So genetic code and translation. So this is the last video class of this unit, because we already finished the genetic code, the first subtopic, and that structure of tRNA, deciphering of genetic code, Obel hypothesis, and protein synthesis, in which you have learned about the activation of amino acids and ribosome structure, initiation, elongation, and termination of protein synthesis. Only we have the last subtopic, that is post translational modifications. In that also only phosphorylation and glycosylation, very small topics, and inhibitors of protein synthesis. So we have a list of inhibitors which inhibits the translation process both in prokaryotes and eukaryotes. In case of replication and transcription also, we have learned about some of the inhibitors that participate in those processes. They act as drugs, antibacterial drugs. Okay, so now let us quickly revise the earlier topics, and then we will go into the final subtopic, that is post translational modifications. Okay, so let us start with the genetic code. As earlier in earlier classes we have discussed elaborately about the genetic code. The genetic code definition is, it is the relationship between nucleotide sequence of mRNA and the corresponding amino acid sequence in a polypeptide chain. So that means genetic code is nothing but this table. Okay, in this table what we observe is that 3 base sequence is called as a codon, and that codon codes for a specific amino acid. So this is the relationship. So what codon is coding for which amino acid, that relation is called genetic code. So suppose UUU codes for phenylalanine, so that means UUU is coding for phenylalanine, so that is the genetic code. So here all 20 different amino acids are coded by 61 codons. So that relation between the codons and their corresponding amino acids is called the genetic code. So here in transcription we have already learned about how the transcription takes place, and from the transcription we will get a mRNA molecule, okay, one mRNA molecule. So this is the mRNA molecule. So from the mRNA molecule, what happens, each 3 nucleotides acts as one codon, and that codon specifies a specific amino acid. Okay, so this way, like AUG codes for methionine, like that. So this is what the genetic code, and the features of genetic code, these are some of the basics of genetic code. And Uh, that first thing is triplet codon. So three nucleotides or bases acts as one codon to code for one amino acid. So therefore, we have 64 possible codons, and among the 64 codons, 61 specifies 20 amino acids and 3 are stop codons. Code is universal, so the code is same for viruses, bacteria, plants, and animals. So that table is universal. Okay, so UUU codes for phenylalanine in all the different organisms from simple to complex level. Okay, so code is without punctuation, so there are no gaps left between any two successive codons. Non-overlapping, the codons are read in non-overlapping manner. Okay, genetic code is unambiguous, non-ambiguous, so that means there is no confusion in the genetic code. So one codon always specifies that amino acid only. So based on the conditions, or different conditions are in suppose in different organisms that may not code different type of amino acids. In all conditions and in all organisms, the, the code always codes for the same amino acid. And the genetic code is degenerate. Genetic code is degenerate because 61 codons coding for only 20 amino acids. So that means more than one codons are coding for a single amino acid. So that kind of codons which codes for the same amino acid are known as synonymous codons. Okay, so this degeneracy is explained by the Obel hypothesis. We will look at about that later. Start and stop codon, we have AUG as start codon, and UAA, UAG, and UGA are the stop codons. Okay, genetic code is polar. It always will be read from 5 prime to 3 prime direction of mRNA. So the codons are on mRNA. So that mRNA is read from 5 prime to 3 prime direction. Okay, but not in the other direction. Codons and anticodons. The codon in mRNA are recognized by the anticodon present on the tRNA. So tRNA is the adapter molecule with its anticodon that recognize a specific codon in mRNA and specify a specific amino acid. So that is why the genetic code relationship uh, is present in the genetic tRNA molecule. So tRNA is the actual translator which reads the codon with one end and specify the corresponding amino acid with other end. Okay, so now this is the genetic code, and you can see all these features in this table. 3 bases acting as one codon, and we have synonymous codons for many amino acids, and stop codons are there, and start codon AUG is methionine. Okay, so all these things you can see here. And genetic code is deciphered by 3 experiments. First experiment is the use of homopolynucleotides. So homopolynucleotides like poly U, poly C, and poly A are synthesized artificially, and they were used as mRNA molecules in self-retranslation system. So poly U, when they used poly U as mRNA, they synthesized polyphenylalanine. So therefore UUU codes for phenylalanine. Like that, CCC codes for proline, and AAA codes for lysine, and poly G cannot act as mRNA because it forms a complex structure like tetraplexes. Okay, use of trinucleotides. This is also known as ribosome binding assay or filter binding assay. So using this, here a trinucleotides 64 trinucleotides were used to bind specific tRNA to ribosomes. If the tRNA carrying a specific amino acid is specific to a particular codon, so that binds to the ribosome and it forms a complex, a ribosome, trinucleotide, and tRNA complex. So that tRNA will be labeled with an amino acid, radioactive amino acid. So when we pass through the solution, pass the solution through filter, the ribosome will be retained on the filter, and so that we can observe by using autoradiography. So in that way, whatever tRNA binds, whatever tRNA binds on the to the ribosome, that will be identified by the radioactivity. So that is known as filter binding assay. Using filter binding assay, around 50 amino acids, 50 codons have been elucidated. Using first experiment, only 3 codons have been specified. Okay, so Hargobind Khurana used copolymers and synthesized defined repeating units, repeating copolymers as the artificial mRNA molecules, and synthesized peptide molecules in a cell-free translation system. So therefore he is able to identify um, the remaining amino acids, remaining codons, elucidate the remaining codons. So these three experiments have led us to establish the genetic code table. And in case of the structure of tRNA, Robert Holly is the scientist who first isolated the East alanine tRNA, and tRNA is a small RNA molecule, single stranded, but it folds upon itself wherever it finds the complementary sequence, internal complementary sequence. It folds upon itself and form such kind of stem and loop structures. And we have 4 arms. First arm is anticodon arm, and that anticodon arm that carries anticodon, which is essential for the binding of codon on mRNA molecule. So on the opposite end we have the 3' end, and the end sequence is CCA sequence, that is common for all the tRNA molecules. At the end we have 3' hydroxy group, to which the specific amino acid will be acylated by a specific enzyme known as amino acyl tRNA synthetase. We will discuss about that later. And we have 2 more loops, DHU loop, dihydrouridine loop, and TCC, okay, pseudouridine loop. So now extra arm is also there in case of some of the tRNA molecules which are slightly larger than the rest of the tRNA molecules. And so this is the clover leaf structure, 2-dimensional structure. When we write it, you will get a clover leaf style like structure, but in fact in 3D structure it is observed as a twisted L shape. And that is about the tRNA molecule and Obel hypothesis. In the Obel hypothesis, the first actually Crick proposed 4 relationships to explain the degeneracy in genetic code. Okay, so that 4 relationships are called Obel hypothesis. In Obel hypothesis, the first 2 nucleotides, first 2 bases in the codon and last 2 bases in anticodon interacts with each other by means of strict Watson-Crick base pairs. Okay, so most of the coding specificity is conferred by these 2 base pairs only. So whereas in case of the third one, so this third base in codon is known as Obel base, and this Obel base interacts with the first base in the anticodon. So that is not completely specific interaction, because there is some flexibility between these 2 bases, and because of that flexibility, the anticodon, the base in the anticodon, the first base in the anticodon may interact with more than one different bases in the codon. So therefore more than one codons may be recognized by only a single codon, single base. Okay, so that was explained in this table. If C is there here, then it interacts with the G here only. If A is there, only one codon having U as the third base. But in case of G, it recognizes two codons. If it is U, it recognizes two more codons, and if it is I, it can recognize maximum three different codons. Okay, so that third base, uh, either it may be C, A, or U, whatever it may be. So that interacts with the I. So that is what the genetic obol hypothesis. So based on this, minimum 32 tRNAs are required for to specify 61 codons, and so that is what the obol hypothesis. And translation process. So in the translation process, we have four stages, important stages. The first stage is activation of amino acids, and the second one is chain initiation, third one is chain elongation, and fourth one is chain termination. In the activation of amino acid, the amino acids must be acylated to the tRNA, their specific tRNA molecule. So this is known as activation of amino acids. So here we require 20 different amino acid tRNA synthetases to acylate 20 different amino acids to their corresponding tRNA molecules. Okay, so first of all, in the mechanism of this reaction, amino acid first um, react with ATP, and it forms a amino acid AMP. So that amino acid now is it has increased its energy, so that it becomes a high energy compound because of the bond between amino acid and AMP. Okay, so now this AMP is simply replaced by the corresponding tRNA, the 3' hydroxy of the tRNA. So therefore, the amino acid is acylated onto the CCA end of the tRNA molecule, and we will get amino acylated tRNA, which is also known as a charged tRNA. So based on the amino acid name, it may be called, like suppose it is the serine amino acid, so then it is seranyl amino acid tRNA. If it is alanine, then alanyl amino acid tRNA. Okay, like that. Methionine, suppose if it is methionine amino acid, then it is methionine tRNA. Okay, so that kind they are charged tRNA. So all the amino acids are attached to their specific tRNA molecules. So once we have the charged tRNA molecules and functional mRNA molecule and ribosomes, and then it starts the translation process. Okay, so in case of ribosomes, we have 30S and 50S type of small subunit and large subunits respectively. In prokaryotes, small subunit is having rRNA, that is 16S rRNA, and 50S ribosome contains 5S and 23S rRNA molecules. Okay, so now during the initiation process, an initiation complex will be formed. Okay, formation of initiation complex takes place. So that is the outcome of initiation of protein synthesis. Initiation complex contains here, so it contains first one the ribosomes, mRNA, and initiator tRNA, that is formal methionine tRNA, and initiation factors IF1, 2, 3, and GTP and magnesium. So during and of course one more thing is the ribosomes are having three different sites: A site, P site, and E site. Okay, so now what happens? The 30S subunit binds IF1 and IF3. Okay, so now IF1 and IF3. IF1 that physically blocks the A site, and IF3 facilitate the binding of mRNA to the 30S subunit. So now that initiating tRNA comes and binds to the P site only, because A site is already blocked. Only P site is empty, and E site, there is that E site doesn't exist in the small subunit. It is largely confined to only large subunit only. So now we have no large subunit, so simply we have P site that is empty. So therefore in the P site the formal methionine tRNA comes and occupies that site by the help of IF2 GTP. Okay, so and the AUG is rightly placed in the P site, which is guided by Shane-Dalgarno sequence. Shane-Dalgarno sequence is a polypurine sequence. That polypurine sequence guides the mRNA in such a way that AUG is rightly placed in P site. Okay, so in case of finally that large subunit comes and binds to this pre-initiation complex to form initiation complex. So now the GTP is hydrolyzed from the initiating tRNA, and as well as all the three initiation factors F1, 2, 3 comes out. So now we have ribosomal complex, mRNA, and initiator tRNA, and now A site is free because IF1 came out. So and during the elongation, the second codon, now it is in A site, and the corresponding tRNA is brought into this A site by the EFTU GTP complex. Okay, so once it binds the A site and GTP is hydrolyzed and EFTU GDP is released. Okay, that is again generated back to EFTU GTP, in which the EFTS is involved, another elongation factor, and GTP. So now this is the first stage in the elongation, that in which the amino acid tRNA binds to the A site, and the second stage is peptide bond formation due to the presence of 23S rRNA in large subunit that catalyzes the formation of peptide bond between the two amino acids which are present on the tRNA molecules. One tRNA is in the P site and another in the A site. So the amino acid from the initiator tRNA, it is cleaved off, and that forms a peptide bond with the amino acid on the tRNA which is at A site. Okay, so now P site is having a discharged tRNA. So and the third step in the elongation is translocation, which requires EFG, which is also known as translocase, an elongation factor. Because of that elongation factor which binds GTP hydrolytic energy, it translocate one codon ahead of the mRNA molecule. So therefore the all the tRNA molecules just shift their positions forward. So now that initiator tRNA enters the E site, and the dipeptidyl tRNA enters the P site, and the A site is now ready to take another amino acid tRNA. Okay, so this cycle takes place for several times until all the codons have been read. Okay, so once the translation is finished, and so that is marked by appearance of a termination codon in the A site. Once the A site is occupied by the termination codon, there is no specific tRNA molecule which can recognize that termination codon. So there is releasing factors, three different releasing factors, and so those releasing factors bind to the A site and cause the dissociation of the entire complex, and polypeptide chain is released, and all gets dissociated, and they are ready to take another round of translation. So this is what the translation process in prokaryotes. So once the translation process is finished, so here once the translation process is finished, what happens? We will get a polypeptide chain. So the polypeptide chain is like that. The first amino acid of the polypeptide chain, newly synthesized polypeptide chain, will be formyl methionine, because the AUG is the first stop initiation codon for every mRNA molecule. Therefore, the formyl methionine is the first amino acid. Okay, so now we have a length of polypeptide chain, and in case of eukaryotes, the first amino acid will be only methionine, not the formyl methionine. So unmodified methionine is the first amino acid in the newly synthesized eukaryotic polypeptide chain. Okay, but when we look at the original proteins, the original functional proteins, when we isolate them from cell organelles or different sources, and if we study the amino acid sequence, in most of the cases, of course in all the cases, no, the first amino acid is not at all the methionine. So that means there you don't find the methionine as the first amino acid. So what is happening there? What about where does it go, that methionine, the first amino acid? So that means that amino acid, sorry, the polypeptide chain, which is synthesized during the translation process, that undergoes some of the processes, they are known as post translational modifications. The same thing we have observed in case of transcription also. So once the transcription is finished, you will get a primary transcript that undergoes post transcriptional modifications. Because of that, we will get a mature mRNA molecule that undergoes the translation process. So in case of eukaryotes, we have three post transcriptional modifications. So first one is capping, 5' capping, second one is poly A tailing, and third one is splicing. So in the same way, we have various different types of post translational modifications. Post means after, translational modification means after the translation, after getting polypeptide chain, that polypeptide chain undergoes various modifications to form a functional protein. Okay, so that is why they are called post translational modifications. We have several different kinds of post translational modifications, and so here we have three important modifications we are going to see in this lesson. They are proteolysis and glycosylation and phosphorylation. Three are basic post translational modifications that will be taken up by each and every polypeptide chain synthesized, mostly in case of eukaryotes. Okay, in most of the prokaryotes, there are very little post translational modifications, but in eukaryotes, it will modify a lot to form different kinds of proteins, and the functional protein and newly synthesized proteins are almost um, uh, they are not similar in most of the aspects. Of course, the amino acid sequence is similar, but there will be many more modifications which ultimately affect the structure and as well as function of the polypeptide chain or the protein. Okay, so the basic uh, modification is removal of the first amino acid, that is methionine, from the N-terminal end of the polypeptide chain. Okay, so that is what the basic thing. So first, as soon as the polypeptide chain is synthesized, the methionine is cleaved off from the N-terminal end of the polypeptide chain. Okay, so now when we discuss about the proteolysis, the name itself explains that polypeptide chains, some of the polypeptide chains undergoes proteolysis. So they undergo some breakdown process in which a functional protein will be formed. Okay, we will get a functional protein after the breakdown of protein in a specific manner. Okay, so that is what we will remove we will discuss in this case. And so some of the enzymes, like in case of mm, enzymes like digestive enzymes, we have some syllabus of zymogen activation in enzymology. So where some precursor inactive proteins are synthesized, which are completely inactive. So in certain conditions, the proteolysis takes place and that inactive protein becomes active. So that kind of proteolysis will activate the inactive proteins. Okay, so here the best example to explain this proteolysis as opposed to translational modification is the proteolytic cleavage of insulin. Okay, so in case of insulin is a peptide hormone which contains two chains. One chain is the A chain and another chain is B chain. So this is A chain and this is B chain. Okay, so this A chain contains 21 amino acids, whereas B chain contains 30 amino acids. Okay, finally totally we have 51 amino acids in insulin. So two chains. So in that case, in fact, it must be the translation system must synthesize A chain and B chain separately, and after that it should combine the A and B chain with by means of the interchain disulfide bonds. But that is not the case. In this case, only one large polypeptide chain is synthesized, and that undergoes proteolysis to form A chain and B chain. So now we will see, we will look at this diagram. You have a very long chain, polypeptide chain of precursor protein, that is pre-proinsulin. Okay, pre-proinsulin. This is having a signal peptide. Okay, this signal peptide is not going to be the part of the active insulin. Okay, so this only for the delivery of the protein from the synthetic site to outside of the cell, secretion of the cell. Okay, secretion into the circulation, signal peptide. So that is again a vast area to discuss. And apart from that, we will have a C chain along with B chain and A chain. We have a C chain, a connector chain between A and B. So now what happens, once we cleave the signal peptide from this pre-proinsulin, we will get a proinsulin. The proinsulin now is having the B chain, extra B chain. So now this forms this kind of insulin structure, and the proteolytic enzymes will cut here and here and remove the C chain. They remove the C chain. Once the C chain is removed, you will get A chain and B chain, both are bound by the interchain disulfide bonds. So we will get only 21 amino acid A chain, 30 amino acid B chain. Each are in connection with each other by means of the interchain disulfide bonds. So now so that means it is synthesized as a single large pre-pro protein, and because of the proteolytic cleavages, it forms the functional insulin, which contains two subunits, but in fact they are not the product of different genes. They are the product of a single gene. single mRNA molecule. So and next one is second one is glycosylation. So in the glycosylation, this is very important modifications for the targeting of the proteins to their target or target sites. So once the proteins are synthesized inside the cytosol, they must be targeted to different cell organelles, or them otherwise they must be targeted to different organs through the circulation. So that targeting requires some kind of markings. So that marking uh, can be utilized for the targeting of proteins. So in that case, uh, the proteins bind some of the sugar moieties, carbohydrate moieties will be present on specific amino acids on the on the polypeptide chain. So this is what the glycosylation. So these carbohydrate moieties can be added to the polypeptide chains. So we have two types of glycosylation process. First one is N-linked and second one is O-linked. N-linked means, so if you have aspargine amino acid, so on the N, on the nitrogen of the aspargine, a sugar moiety will be linked. In most of the cases, that is N-acetyl glucosamine. Okay, so that is linked to the nitrogen of the aspargine amino acid residue. So that is N-linked glycosylation. In case of O-linked glycosylation, you will get that carbohydrate moiety on the oxygen atom of amino acids which contains hydroxy groups like serine, tyrosine, threonine. So these amino acids take up the carbohydrate moieties and they are called O-linked glycosylation. So the final one is post phosphorylation. So as we all know, we have already discussed or came across with the regulation of regulation of metabolic process, okay, in which some of the proteins, some of the enzymes are activated and deactivated based on the phosphorylation and dephosphorylation. Okay, protein kinases are the enzymes which phosphorylates the proteins or enzymes and either activate or deactivate. Okay, in the same way, some phosphatases also there which will remove the phosphate groups from those proteins. So one of the example is shown in this slide. So phosphorylase enzyme, kinases enzyme phosphorylates the proteins. Okay, and phosphatase enzymes release the phosphate group from the proteins. So suppose if we take the example, some glycogen phosphorylase enzyme, what happens that glycogen phosphorylase will cut glucose molecules from the glycogen. Okay, so that glycogen phosphorylase, when it is in phosphorylated form, that is in the active form. When it is dephosphorylated, it becomes inactive. So that same protein, the same enzyme will be in active form once it is phosphorylated, and it will become inactive once it is dephosphorylated. So that phosphorylation and dephosphorylation is the key to switch on and switch off those kind of enzymes based on the cellular need. Okay, this is what the phosphorylation. And the last topic is inhibitors of protein synthesis. We have plenty of different protein synthetic inhibitors which inhibit protein synthesis process both in prokaryotes and eukaryotes. Some inhibitors are exclusively act on prokaryotes and some will act on eukaryotes. okay, based on the differences in the components used in the translation process. Okay, we have some of the examples like puromycin, emitin, and anisomycin, sodium arsenate, and herring tonic. So all these drugs, protein inhibitors, are used as inhibitors for the synthesis of proteins. Okay, so this is about the entire translation unit, where we have studied about genetic code, experimental evidences to elucidate the genetic code, and tRNA structure, ribosome structure, Oval hypothesis, and activation of amino acids, amino acid tRNA synthetases, and initiation, elongation, and termination, and finally post-translational modifications and the inhibitors of protein synthesis. Okay, thank you one and all.